Good morning, thank you. I'm not sure about 2004. I actually announced in, I think, 2008 that I was retiring, whatever that means, from my position as the chief scientist of the Roche group, which includes obviously Genentech, Roche, Chugai Diagnostics. First, thanks very much to Ernst and colleagues for organizing one of the most interesting dialogues and discussions I've attended on this subject. Extraordinarily important. I, like many of you, have been very interested in two types of questions, mostly about biology and medicine. The first question is, how does it work? Which is an extraordinarily interesting question, in which many of us have spent a lot of time trying to understand how bits and pieces of biology, some large, some small, some related to medicine, some not. How do they work? The other thing, the other distinguishing feature for many of us is that we are also interested in, having understood how something works, to actually put it into practice, make it useful to people in need. And if you do this in medicine, it usually means some kind of diagnostic or some kind of therapy. So what I wanted to talk to you about today is my personal perceptions and prejudices on what we need to do to create more effective therapies. Because as I will show you, I think we are approaching a crisis point, and we need, in order to come out the other side of this crisis point, we need some coherent action taken, not just in the industry, not just in academia, but throughout society. And I want to try and give you a sense of that. Now, the first thing to say is some of us anyway, talk about personalized medicine as if it was brand new. There is a great deal about what we do today that is brand new, but I believe that the concept of a clinician treating each individual as a separate individual is not terribly new. I would suggest that if you go back 10,000 years in the woods around here, you would have found somebody in society who was creating different spells and different potions for each individual. If you look at the size of the herb garden that they would have had at the Abbey 500 years ago, you will find that was a very significant collection of plants, not to mention the ones they brought from outside. So the concept of personalized healthcare is not new, I would contest. We have, I believe, taken, in some senses, a pace backwards with some elements of what I would call evidence-based medicine. And we've taken a pace backwards because the evidence-based medicine was based on larger and larger populations, which means that the therapeutic solutions we proposed to individuals were probably even less. If you look at the cancer guidelines, for instance, in the US, they are even less individualized than they might have been, certainly for early cancers, let's say even 10 or 20 years ago. And so I think this is a challenge that we need to address even more rigorously. As I said, what hasn't changed is, I believe, the intent to be personalized. What has changed, as we've heard beautifully from George and Rudy and others, is the technology that is exploding even more dramatically in biology than any other area ever seen. I would just point out that I believe today, so here it says tomorrow, patient management based on individual computer predictions, which is what we just heard. I believe that is already true in cancer. It is already impossible to practice state-of-the-art cancer without an algorithm or a program that actually analyzes all of the clinical information if you want to make good decisions. And I will show you what I mean. Now, the crisis that I was referring to, this is one way of illustrating it. And this is, as you can see, this is the number of new medicines registered, in this case in the United States, divided, or at least not divided by, that's the bit that comes down from 53 in 1996 down to 18, actually it's now up to 20 for this year. But what you also see is the R&D spend, and you can multiply that by about 2.5 to get the world spend. And I think there are two important things. One is this is completely unsustainable. And that is why, for instance, the market caps or the PE ratios of pharmaceutical companies are less than those of cement companies, because cement companies have a future. Investors and many of us believe that pharmaceutical companies in their current form do not have a future. There is huge value to be brought to patients and healthcare systems, but it cannot be brought with our current way of thinking. And when I say ours, I don't just mean pharmaceutical companies, biotech companies, I mean all of us, including healthcare providers. The other interesting thing you can see is for the first time ever, you see a decline in the investment in the creation of new medicines. Most of that is clinical studies, but you will also personally be aware of the closure of many sites. This is an accelerating issue, and what that will mean is that many of the clinical studies we've been accustomed to see funded by companies actually will no longer be funded like that, and therefore many of the discussions in this meeting about how else do we generate clinical information, I think are extraordinarily important. That's the downside. The upside is that we, mankind, which is everybody, have been extraordinarily successful in creating a very large range of mostly relatively safe medicines. 7,500 are, I'm told, on the Swiss formulary. If you go anywhere near a hospital in Switzerland, you probably get about 20 or 30 of them on the first day. I speak from personal experience. But I think, and this has been alluded to by a number of speakers, and I speak from personal experience, the rationale used to say, I have a target, and here are the patients, let's put this molecule that we've just made into these patients, is extraordinarily tenuous. Take it from me. It's a sort of it's not exactly a fairy tale, but pretty close. And what this means is we do have 7,500 agents already that do all sorts of weird and wonderful things. Many of them do several things at once. And what is at least as important as the creation of new medicines, as we heard, is the understanding of what else these medicines do. How do you translate somebody's side effect into a therapeutic benefit for a completely different disease? And I think that's a huge and very important opportunity. This will not be funded by the pharmaceutical companies. And so we need to find other ways, or change the inducements, change the reasons for investment in society. Remember, companies, they're only an instrument of society. Patents, they're only an instrument of society. We could change that. There are lots of reasons for the graph, for this graph, but I believe one of them is the way in which many of us still run clinical trials. It's changing, but much too slowly. As you see, and you know, most drugs only work work really effectively in a rather small proportion of the patients as we define them. When you run a clinical trial in an indication, you are forced by current accepted wisdom to take basically a population of patients that is undifferentiated, because that's all we know, and that's how we classify diseases. The trouble with that is when you run your brand new molecule that may be stunningly efficacious in 20% of the patients you've included in your population, and you average out the efficacy, that looks rather like the generic molecule that's slightly less effective in 30%. And currently we have often no way of saying, is the 30% affected by the generic the same as or different from the 20% who are highly well treated by the new medicine? For that reason, it's one of the major reasons, 50% of phase 3 studies have been failing over the last few years. 50%. If you think a phase 3 study costs about 500 million, sometimes more, you can see that's completely unsustainable. That is not going to continue. And so I would say it is a very serious crisis. We may be too late, actually, for this generation of companies. But that's the challenge. We have to redefine disease such that when we run a study, we know much better who to give it to. And there are other safety issues. Now, the interesting thing about safety data is that safety data is collected worldwide, often not very effectively, but it's connected in the absence of information about the efficacy of the medicine in the population from which the safety is being gathered. Now, the efficacy might be much more, or it might be much less. There might be very little efficacy, actually, in practice, compared to the clinical trials, and there's some data to say this. So it's another argument for saying we have to completely re-engineer the way in which we generate clinical data. This, I have to say, is extremely controversial. If you bring this kind of perspective to a group of practicing clinical trials experts, they will say, nonsense, go away and run a clinical trial. So I think we have some work to do. And just to underline the point, I think this was, it was obvious, obviously, to T. Lewis, who's a clinician in London in the last century. It was obvious to T. Lewis that this issue that we're discussing was serious, even in 1944. And he said, as you can see, diagnosis is a system of more or less accurate guessing in which the endpoint achieved is a name. These names applied to disease come to assume the importance of specific entities, whereas they are, for the most part, no more than insecure and temporary conceptions. And I said, this is a very clearly said. I have to say that my reason for being interested in personalized medicine was because about 20 years ago, it was very clear that the graph I showed you was going to happen, and it was equally clear that without understanding to whom we should give our medicines, we were not going to make any progress. I think it's useful when we're talking about personalized medicine to divide our thinking into two different categories. One is stratified medicine, which is a bit what I've been talking about, which is new classes of patient subgroups. And the other is personalized, really personalized medicine, where individual and often novel combinations of agents are given to patients on the basis not of significant clinical data, but on the basis of if you like, some kind of molecular genetic rationale. And these are very different issues, because you can, of course, run stratified medicine clinical trials, and many of us do. It is much, much harder to generate the clinical data to support personalized medicine. It is possible. So pharmaceutical companies will help us and are helping us with the first. To get to the second, we'll need, as I said, a completely different paradigm. Let's start with stratified medicine. And I just have a few thoughts about how we might start to think differently. And some of us, clearly, as many of the speakers have actually alluded to this. So this is an article looking at the response of olanzapine haloperidol. It's by Kshitij Kapoor and his friends in London, in schizophrenia. And the first thing you might say about schizophrenia, actually, it's normally classified as a psychiatric disease. However, there's another perspective that said, well, just a minute, maybe some part of schizophrenia is neurodegeneration. And maybe schizophrenia and neurodegenerative diseases have mechanisms in common. But be that as it may, what the important point of this study is that if you look at if you classify patients into response, these are the individual patients and these are the averages, you can classify them into three, or they have classified them into four types: non-responders, partial responders, responders, and dramatic responders for these two drugs. Now, the important thing about this is that about 90% of the data that says these drugs work in schizophrenics comes from the dramatic responders. This is only about 10%. So actually, when we say these drugs work in schizophrenia, that is actually not a terribly helpful statement. It's some probability that it will work, and in this case, not an incredibly high probability. Of course, I understand there are difficulties with defining response and all the rest of it. But one of the most important points I want to make, and I think this is now changing, is about deep phenotyping, and phenotyping. A drug response, a dramatic drug response in a patient, is one of the best molecular markers we could possibly have. What is extremely interesting is this information is very difficult to get hold of. I was saying during the break that I've come across 2 large biobanks of diabetes patients. And in one, the weight was known, but the BMI was not possible to know because the height wasn't known. And in the other one, it was not possible to have any information on whether or not patients respond to metformin, a very common diabetes drug. Now, if you don't have these kind of informations, then it was rather a waste of time collecting the samples in the first place. And so I think a great deal, I'm sure this is happening, but from my perspective, as George said, and others have said, we need the most amount of information we can possibly get. And until we get that, and a particular drug response as one of the best molecular markers around, I believe we're not going to make much progress. And that's one of the reasons why we've spent hundreds of millions around the world on biomarker studies. And most of that, with the exception of Rudy's, most of that has been, in my view, a waste of time. And the reason it's a waste of time is because we're sort of asking questions about large populations, and therefore we get not much of an answer. Alzheimer's. So this is an area, I'm just extending what was said a little earlier this morning. Until recently, until about 2 years ago, we basically had 4 risk factors, ApoE4, that we just heard about, and then the familial ones. And all of this was used to say, actually, Alzheimer's is a disease of amyloid. And I've heard in the last month, I've heard a number of people standing up say, amyloid is the only CNS disease where we have absolutely clear pathology, we know exactly what's going on, and we're going to fix it. I have a contrary position, partly because I've been personally responsible for spending at least 70 million Swiss probably, on doing the experiment of removing amyloid from patients' brains using antibodies, and there are a number of other groups who've done that. And that, unfortunately, did nothing for cognitive performance, and there are lots of reasons why that might be. However, what I think is extremely interesting is that recently, in the last couple of years, there have been a whole slew of new genetic risk factors. And if you say, what does this mean? I don't know if you would agree, Andres, but if you say, what do these mean? They say something like, well, actually, for common Alzheimer's, it's probably more about membrane metabolism or some kind of metabolism and maybe inflammation and some other things. My point is that instead of fixing on the first hypothesis that we came up with, which is, by the way, well over 100 years old, we need to continue to evolve our understanding of disease and to try and redefine Alzheimer's in the context of new information. I have to say there's a big disconnect, particularly in, I would say, in academia, actually, between the thinking of people who are fixated on, let's say, amyloid or whatever, and the emerging medical data. And that's something this bridge we need to bridge. We need to bridge better. Just a cautionary tale from diabetes. So diabetes, for 5,000 years, has been recognized by sugar in the urine and more recently in the blood. And so a lot of us thought, well, if we just fix that, then we probably fix diabetes. And this is a lovely story. This is done by the molecule here, was made by my colleagues in the States a number of years ago. And then that story is very nice. So Moody 2 is a familial early onset early onset diabetes gene. And when you've got it, you have a high probability of getting diabetes. And it actually changes genetically the structure of glucose kinase. And so we reasoned, well, if we can reset the mutated glucose kinase back to wild type, everything should be fine. Should have a great diabetes medicine. Yes, we could reset both the mutant and everybody's glucose kinase back to normal. And it worked. It actually did normalize glucose in normal diabetics. Interestingly, it turned out, despite this genetic evidence from a rare familial thing, it turned out that glucose, as actually Emmanuel was telling us, glucose is actually kind of so far down the chain of diabetes that modulating glucose is, with the exception of preventing death and coma, which is a rather late stage, is actually not a useful mechanism. It's not a useful way of thinking about diabetes. And it has to do with many other organs, not least the brain and the gut. And that is where the solutions are being found from. There's just a cautionary tale about genetics and targets that I think is, for me anyway, is an important learning experience. Let me now come to cancer. Of course, cancer is if you like, the poster child, I would argue, of personalized medicine, because it is essentially a genetic disease. Because obviously cancer evolves because it mutates and therefore is able to evade and grow. And this year is the 10th anniversary of this diagram, which is the Hanahan and Weinberg diagram, and it basically shows our collective thinking about cancer. And my colleagues and I, not just in Roche, Genentech, but around the world in academia, we've all thought of cancer as a disease of division, growth, and death. That's how we thought. We thought about it from the perspective of cancer. And so we made a lot of molecules. And there are, as somebody said, there are 1,000 molecules, 1,000 cancer molecules in development in the US. It's an awful lot of molecules. So actually, there are two things I'd like to conclude from that. One is actually making the drugs no longer seems to be difficult. That seems to be easy. Most people can do it, or not most people, some people can do it in their kitchens. That would appear to be the case. Very successfully, safe, targeted, all the rest of it. The challenge is we don't know who to give it to, which I'll come back to. The other challenge is that actually when you try these drugs in patients, you kind of get what you'd expect. In other words, if you develop a drug against cancer growth, what happens when you give it to a patient? Well, the cancer growth slows down. It doesn't disappear, it kind of slows down. And if you use it as a single agent, the cancer gets resistant and goes on growing. And so I would argue, again, that our thinking about cancer is far too limited. And recently, I think somebody, I can't remember who said this, recently there have been some really dramatic developments from taking a completely different perspective on cancer. If you say cancer is actually, essentially, mostly a disease of immunoevasion, it's about evading the immune system, and we should worry much more about the relationship between the immune system and the tumor, and that's how we will fix it, we might cure people, and that's actually what we are starting to see with a number of systems, tumor-tropic viruses, gene therapy in T cells, soluble T cell receptors. That's what we're starting to see. It's just another example of how our thinking is too constrained by our original classification of the disease. And by the way, I forgot to mention there's an interesting paper that shows quite good cognitive improvement in Alzheimer's through nasal insulin. So I wouldn't like to claim that Alzheimer's is a form of diabetes, but you do have to ask different kind of questions from the kinds we've been asking. Let me just show you one example of what happens when you do know which patients to give it to, and this is a program that I was very proud to be associated with, and that's the new data on BRAF. So this is a partnership with Plexicon, and you can see BRAF mutation occurs particularly in melanoma, but actually with a reasonable frequency in many other cancers. Interestingly, this is in 15 days. If you get the right drug and you give it to the right patient, you can see a dramatic reduction this is FPET, a dramatic reduction in the cancer. So this is really making a huge difference. And you can see this in terms of survival, progression-free survival. Brand new molecule, amazingly efficacious. What's the downside? And by the way, just approved by the FDA, rapid approval, because in the clinical development was both a diagnostic and the medicine, both submitted at the same time. All works. This is one of the first examples. Pfizer, I think, has or will have another example, but this is one of the first examples of this sort of co-development. Herceptin and actually Gleevec were early, but that's taken about 15 years to get the next examples. However, what's the downside? The downside is after about a year, or a bit less, all the patients got resistant. And what that means is, if we want even to control the tumor, we will have to give drugs, most likely, in combination as we treat HIV. And that poses a big problem. Just in passing, So clearly, and there are a number of cancer drugs, as you know, that require mutations. The ability to run this type of molecular pathology is somewhat patchy. There are centers in the US, there are not too many centers in Europe, or in Switzerland actually, who actually do this on a routine basis for all patients. But we're kind of getting there slowly. I just thought it's worth noting this is from a presentation given by Fabio Calvo, who's the Deputy Director of the Institut National du Cancer. And actually, 4 years ago, the French set up an integrated cancer molecular screening system, 28 centers, all integrated. And that is one of the largest populations that I know of that is routinely screened for cancer. Let me now move to personalized. How do we get to this? As I just said, BRAF isn't much good by itself. It needs to be used in combination with something. And, I just want to show you, it's not that patients don't get a lot of different drugs. This is really impossible to see. This is from 509 ovarian cancers, patients in Finland treated with 124 different drugs and combinations. And unfortunately, you can't see it. You still can't see it. There would be lines, so each patient would be represented by a line that goes through all of these things, and then they end up living today or dead. My point is that today, doctors do give a whole lot of different drugs. However, the evidence they use for that doesn't bear much relation bears relation to the state of the patient. That's very good. It doesn't bear much relation to the molecular pathology of the cancer. Now, one of the things that I did when I retired from Roche about just over a year and a half ago was to join the board of a company called Caris. Caris is a very interesting company. It does molecular pathology of nearly 1 million patients every year. So it's one of the histopathology for nearly 1 million patients. So it's one of the largest histopathology organizations in the world. It also does molecular pathology. And here we see, this is Target Now, which is actually derived from the work of Dan Van Hoff in Phoenix, who is one of the pioneers of personalized healthcare. And what it does is to take all of the relevant information that you find in the clinical data, that is immunohistochemistry for up to nearly 50 different antibodies. So we're measuring 50 different proteins, which is, by the way, rather different from what happens, if I may say, not too far away from here, where you might get 2 or possibly 4. So that's different. A microarray of all of the gene expression data that is then used to correlate and confirm that. And then, of course, FISH, this is a longer list, and then mutational analysis, and this is a much longer list. So that's kind of okay. We could all do that. What I think is interesting, though, is what happens to this information. There is a team of 6 molecular biologists, molecular PhD biologists, some of them have medical degrees, with a pathologist, a board-certified pathologist, who every day, they read the literature, or they analyze the literature, and they translate the literature into rules that allows the weighing of evidence. And all of this is put into a computer program. And what this program does, what this program does is to assess against published literature with rankings, to say this is important, this is important. It actually gives a prediction of which agents are associated with clinical benefit and which agents are associated with lack of clinical benefit. And this is at least as important for the old chemotherapeutic drugs as it is for the brand new ones. And it gives you, this is a report for a patient with colorectal cancer, it gives recommendations, in this case, irinotecan, because of high expression of TOPO1. It also recommends, for the patient's consideration, the clinician takes the decision, this just provides the data for the clinician, it recommends paclitaxel, not because there's any evidence whatsoever that this works in colorectal, but because it has worked in breast cancer, and there's high expression of SPARC, which is correlated with the permeability of tumors to paclitaxel. So you get the idea. And what I think is extraordinarily important about this is that, for instance, 12% of colorectal cancers have high expression of HER2. So they are completely eligible. They are as eligible, I would argue, as a breast cancer or a gastric cancer patient for at least for consideration. That is completely missed. And then the other side of this, of course, is that in many cancer organizations, Herceptin, just to take an example, is given to patients with decreased P10 because the P10 is not measured. I mean, I would say that was irresponsible to give a drug like Herceptin or any of these other tyrosine kinase inhibitors to patients who already have a reduced P10, which means nothing you do up here is going to affect anything down here. This is today, and my personal belief is that this is essential. Whether it's Caris or somebody else, this type of approach is essential for the effective treatment of cancer. It does bring a challenge, which I mentioned before, and that is, how do you show this clinically? How do you run a study when each patient in your study, if you've got 10,000 patients, then you may only have 3 cases who get a particular combination. And this is a big challenge, I think. It's the relationship. How do we move medicine forward with this kind of thing? And I think what now is being set up is, as Dan Van Hoff has to some extent done, and others have starting to do, is you need to compare not drugs and regimes. You need to move away from that. You need more like in surgery. You need to compare decision protocols. How did we take the decisions? And how did this way of taking decisions benefit patients versus that way of taking decisions? And I am certain, by the way, implemented with this would do 2 things. First of all, it would dramatically extend patients' lives. It would certainly cure a few. I'm certain it would cure a few. And not only that, it would also reduce the cost of cancer care in countries like Switzerland and America, because we would stop giving drugs to people who clearly don't benefit them. We would stop giving, for instance, cytostatics to people, make them depressed, remove their immune system, have their hair fall out for no good reason. So in my title, I said, vision or immediate necessity. In cancer today, it is an immediate necessity. Let me just quickly move on to give, and sorry, this is just to say, how on earth, this is now more than 1,000, how on earth are we going to clinically develop 1,000 drugs, cancer drugs? How is that going to happen? It's not going to happen. And yet, many of these molecules are valuable for somebody somewhere. That's about combination. Just to extend, it's about combination. Just to extend the topic that Rudy brought up yesterday. So I became extremely interested in as many of us are, because of the problem of biopsy and indeed the whole question of how do you measure things. And one of the most interesting developments in the last 10 years, particularly in the last 2 years, has been the understanding of the key role of exosomes. And exosomes, as you you may or may not know, are interesting vesicular bodies that are secreted by cells. And basically, I think where we've got to, at least where I've got to, is to say there are 3 main ways in which cells communicate with each other. Cell contact, in tissues mostly, and the immune system and the brain. Hormones, soluble, all go around the place. And very importantly, exosomes. Exosomes are interesting. They contain a significant amount of proteins around the outside. They contain the master controllers of gene regulation, microRNAs, and they also contain messenger RNAs. And they also get in and out of the brain incredibly easily, as Matthew Wood has recently shown. Next, please. This is a very good review. Next. There's a very good review by Thierry et al., who are immunologists. And you can just see from this, if you maybe can't read it, so exosomes have been shown to release tissue factors, coagulation, thrombosis, inflammation. They maintain immunotolerance. They immune suppress dramatically. They do angiotensin, they do apoptosis, tumor progression. Basically, exosomes have been shown to do a whole lot of things. And as I said, Matthew Wood just created a synthetic exosome that, when injected into the tail vein of a mouse, went into the brain and turned off base 1 in the brain using a tailored microRNA. So they do a lot of things. So obviously the question that we've been asking, and particularly this is the work done by my colleagues or the guys in Phoenix, Arizona, in Caris, is, okay, well, could we use this as a diagnostic? Instead of saying, let's look at everything in the blood let's look at the signals that cells specifically send to other cells and see if we can't understand what they're saying to each other. Next, please. And when you say, well, what do exosomes look like? And they actually are extraordinarily homogeneous. These are from the cell line. Here they are stuck to a bead. They're extraordinarily homogeneous. And actually even appear to have some kind of internal structure, sort of an external structure. You can see the concentration of proteins on the surface of the exosome. Next, please. One other of the interesting things, and there's a number of data from different groups saying, first of all, that people who get sick often have elevated levels of exosomes. Just say, how many exosomes have they got? Some of that is inflammation, but some of it is specific to other cell types. This is just some data from Knowles that says, actually, if you look at exosomes in a set of cancer patients, these are prostate cancer, colorectal, esophageal, whatever, you see a significant increase. So in cancer, people tend to have, not all people, but many people have more exosomes. Next, please. So what what the Knowles group did was to ask the question, if a prostate cancer cell makes exosomes, you could argue that it would have two types of antigen on its surface. It would have prostate antigens, and it would have cancer antigens. And if you find these two antigens on the same exosome, you have some reason for believing that that person probably has cancer. And that's exactly what was done. I don't have time to show you. Yeah, okay, next. And it's in sort of Illumina immunoassay, as you just saw. But I think what is interesting is that using this approach with, in this case, I think this was 7 different markers, it clearly looks like it's possible to tell people who've got cancer from people who haven't got cancer. There are a number of problems with prostate cancer, as I can't remember who said this, as there are with breast cancer, and that is, one of the problems is using existing technology. We have a real hard time saying actually who's got real cancer that's going to kill them and who's just got some benign nodule. So that's a big problem. It's not a technological problem, it's a problem with the classification of patients using our existing tests. I find this extraordinary that you give a pathologist a chunk of prostate or a chunk of breast, and they can't tell you, is this going to kill you in 10 years? Well, sometimes they can if it's metastatic already. But often they say, well, you've got cancer, you better have your prostate out. I mean, whereas actually, particularly if you're 67 or something, that might be a very stupid decision. Anyway, but this is not yet ready for prime time, but I believe it will be sometime the end of this year. And it will do also, an enhancement of this test, actually, I think will give probably even better data than is given by many of the current systems, not least PSA. This is in the same patients. Sorry, it's PSA. That's the thing. Okay, next, please. Now, one of the other things, as I mentioned about exosomes, is, of course, they carry some degree of message. So what you can do is, this is exosomes from colorectal cancer patients, and the important thing is that you can detect from plasma the mutation of the KRAS gene, which I think is interesting. Now, of course, you can do this with CTCs. However, the frequency of capture of CTCs is not as effective as one might like. This is, I would say, close to 100% of the patients you can get this kind of information. Next, please. I think at least as important, or actually probably more important, are the microRNAs found in exosomes, because those are probably the main ways in which cells control other cells. And here, this is a partnership, collaboration we had with Freddie Handy in Oxford. This is a poster this year. And essentially, what this says is that it looks like with more data and more patients, we will be able to tell whether, just from a blood sample, whether or not a patient has metastatic prostate cancer versus normal cancer. And the reason is because, of course, when the cancer goes metastatic, it needs to send out other signals, different signals, to the tissue in which it is planning to grow. If you look at the literature on exosomes, it has gone exponential just in the last 2 years. It was only in immunology 10 years ago. It's gone completely exponential. And so I'm extremely optimistic that I have the next, please. Go back one, sorry. I'm extremely optimistic about the potential for getting really fine diagnostic definition for cancer patients. The breast cancer data, which I didn't show you, looks even better. There are other, Larry Gold in SomaLogic, while he doesn't claim to be studying exosomes, has some very persuasive data on lung cancer. And so I'm really persuaded that in the next few years there will be revolutionary new diagnostics. First for cancer, but in the context of our discussions about Alzheimer's, diabetes, depression, schizophrenia, I believe that looking, as I think George was proposing, looking at what's happening in the serum, in my view, particularly through exosomes, is going to give us a completely different perspective and completely different ways of clustering disease, clustering patients into diseases on a much more mechanistic way than we have been able to do before. Next, please. So I said in the beginning that I think actually the science and technology is going pretty well. Where we get stuck is with some of our inability to think beyond our classical definitions of disease. I think it's very important for all of us to encourage each other to think bravely and more differently. And actually that requires, I would say, rather different kinds of discussions between all of the 6 sectors, all of the 6 participants, key stakeholders in healthcare. And I think it's particularly so, earlier this meeting we saw a slide on the benefits. So I think it's all clear that if we could do this, we would all benefit. That's possibly accepted. What is probably even more important is what are the contributions? How do each of these groups of people need to think differently? And I've been extremely encouraged during this meeting to see quite different types of thinking, particularly in the context of the full engagement of patients, actually to take over, if you like, some part of the role of generating clinical evidence, because I believe that's absolutely essential. Next and last. So just in conclusion, these are sort of my personal recipes for what we, as a society, those of us interested in healthcare, actually need to do. I think all of us need to focus much, much harder on the reclassification of disease. We should become very uncomfortable every time we're offered a cohort of patients, or every time we think of a study that is simply a collection of patients as they were classified. And I believe that is essential in order to create new therapies, I would say, for anybody. So there won't be any new medicines. Won't be too many, and maybe 1 or 2, but there won't be a lot of new medicines until we've solved that. As I've said several times and has been referred to in the meeting, we need a major innovation in how we demonstrate clinical efficacy, which has much more to do with retrospective and prospective analysis of highly detailed cohorts. We need to move on from, so companion diagnostics is now fashionable, just. I believe that is a very limited perspective. We've already gone beyond that in cancer. So companion diagnostic is already an outdated concept for the treatment of cancer patients. That will become so in other diseases. I didn't mention it, but my colleagues in California have some very interesting data on redefining asthma using a serum marker. And they have a medicine that works very effectively in a subpopulation of asthma. So that's where it starts. As I said, as we do this, then we will need, as we do in cancer today, we will need information systems to give the clinician the kind of, as the previous speaker said, to give the clinician the information in an actionable form. And finally, I believe that this is essential to encourage novel partnerships and collaborations between science, technology, and medicine. Many of us here do that. But there is, as I said in the beginning, there's still, for my taste, a significant isolation between the people who work on so-called model systems in laboratories and the people who treat patients. And we need to bring that together. Anyway, thank you very much for your attention.
Thank you for a wonderful talk. You said something early on that I'd be interested if you'd expound on a little bit. You talked about the need to re-examine the current sort of accepted hypotheses within disease or definitions of disease. And I have my own observation, which is that it is necessary but not sufficient for whoever is in charge of a disease to reject the hypothesis for it to be true. And I'm wondering if there's a way that you could formalize sort of a new profession of re-examination, that you could, that you could create something where there was a process of coming in from the outside, and because the information is moving so quickly and we cling so tightly to our theories that I wonder if there was a way that we could create a field of doing that in a rigorous way, that much in the way McKinsey re-examines a company or something like that, that would allow us to challenge and advance knowledge in spaces.
Yeah, I thought you had part of the answer, actually. That's why you're asking the question. One of the, when we were discussing, the 7,500 drugs and how they all have side effects in somebody, but they all the rest of it. One of the wonderful things about the information you're putting together is that allows you, to the degree that we could persuade patients to give a holistic view of their perspective, that then allows us to say how strange, a bit like, for instance, metformin is a very good example. So it's kind of interesting to note that people taking metformin for diabetes got a lot less cancer. And once you know that, then you can go back to the molecular pathway, ACT, etc., and you can say, well, of course they would get less cancer. And if we go back about 30 or 20 years, Pat McGeer was showing us data that said that rheumatoid RA patients who took high-dose aspirin had a significantly lower risk of Alzheimer's. So it's sort of kind of, if we can get to populations, so one interesting thing is, okay, let's look at the aspirin responders in Alzheimer's. Forget about the rest, okay, they got Alzheimer's. But what about the guys that we think should have got Alzheimer's but didn't? That's a completely different population to be looked at in the context of Alzheimer's. So I think, this is difficult to do. A lot of this, one of my reasons for working part-time in Scandinavia is because the Scandinavian medical databases are lifelong, and there is a very great deal of information, and that's why some of the papers showed earlier, actually you showed them, are from Finland, and that's one of the reasons. And so I think that's one place you could start, but I think the kind of things that you and others doing, George is doing, actually is going to take us quicker, perhaps, to the right kind of answers. I don't have an answer, but I think you're doing it.
Another question? Would you like to go ahead?
Thank you very much for your interesting talk. I have one question regarding your first point about the stratified medicine and your comment when you said that 50% of the phase 3 clinical trials fail even though they cost about 500 million. So I was wondering, are pharmaceutical companies trying to figure out, because you also showed these curves about the good responders and bad responders, kind of trying to figure out what is the reason why people respond well, and if they do that, or if they don't do that, why do they not do that?
That's a very good question. So I have to say, I've spent the last 15 years of my life trying to persuade the pharmaceutical industry generally to think differently about their studies. And there are 3 obstacles. The first obstacle is the traditional perspective of the pharmaceutical company that says the more patients there are, the more revenue we will earn, and the more we can pay our shareholders and all the rest of it. That's traditional. Now that is about 70% disappeared. You are still left with a mostly academic industry of clinical trials, actually. So it's a kind of industry, and there are so-called key opinion leaders, and you get and these are the people who run the trials, and they're the ones who take the time out. And until a couple of years ago, and I used to run a lot of these scientific advisory boards, until a couple of years ago, there was a kind of reluctance to think about running a clinical trial with the required amount of biomarkers. So there was even the sort of, well, that's not really, I don't think we can ask them to give these samples, we can't do this, we can't do that. Interestingly, if you ask the patients, like, I mean, we ran a lung cancer trial, and we said everybody in this trial needs to give a fresh biopsy. And the surgeons and patients said, okay, we can do that. But prior to that, the view was you couldn't do it. So I think there has been, it's changing because of the work that we're doing. It is changing, but I mean, there was, if you said I want to do a diabetes study, you got your diabetes population. You might have got mild diabetes or moderate diabetes or severe diabetes, but that would have been it. And so I think there's a reluctance in the medical community. And then finally, there is a reluctance both in the regulators to say, well, I'm not approving that study. You need to have all diabetics in here. Otherwise, you might be killing somebody that you didn't test. So that, again, makes the cost of the trial bigger. And then there's a reluctance, and this is one of our big challenges, there is no funding for molecular diagnostics in healthcare systems. So we are not happy, but we do spend we spend millions and millions on very expensive drugs without being bothered to spend, a hundredth of that on the diagnostic that would say, don't give this drug to this person. And that's a sort of structural thing. So that's 4 I've given you. Is that all right? Is that enough? So there are reasons. Now, they're all shifting, but very, very, very slowly, and much slower than the information that we now have available. And this is, sorry, just one thing. So this Target Now analysis is available to anybody, anywhere in Europe, anywhere in the States. In the States, it's reimbursed. And yet you still find clinicians who will say, even when a patient has run out of options, I met one on Monday, patient had run out of options, failed 3 therapies, ovarian cancer, and this clinician refused to give her the one drug that was indicated by the molecular analysis because he said there is no clinical trial. I met him, and I said, but she's going to die. And he said, yeah, probably. So anyway, reluctance, sorry.
With your proposal that one should reconsider the classification of disease, I was wondering what would be your position concerning also similar studies with rare or very rare diseases. If you take the example of certain muscular dystrophy, one of them gave very, very interesting information on mechanisms which are important for elderly people when they get their muscle atrophy. Yeah. And this type of trying, let's say, to neglect because the cohort is not big enough. The rare disease is possibly a danger, and one should, let's say, take them on the train as well. What do you think?
I think it's very important, and I've actually personally been involved in a number of studies in rare diseases precisely because if you can get the family, and the family is usually happy to participate, and if you can do, in that case it was GWAS, but these days you do a complete sequence, then you are quite likely to come up with at least proposals for existing drugs that might help. So I think it's an extraordinarily good idea. And having done that, I can't remember who said this, somebody said, you know, all cancer is moving towards, everybody's cancer is a rare disease. You know, that's probably true for diabetes. Everybody's diabetes may be a rare disease. And so by studying obvious high-penetrant rare diseases, I believe we are likely also to get insight into some element of more common diseases. Very important.
There was another question in the back. Yeah.
I just had a comment on your last slide where you speak about collaboration and the case you said with the patient who didn't get the single medicine she would need gives you that collaboration. I would add the patients because we in the clinical tend to forget, but again, HIV patients, some tumor patients get very well organized, and MS patients like we've shown in the US are very well organized, and then if we collaborate, then with them primarily without, and accepting that they have a view. Maybe it's not always our view, but they have a view, and it may be interesting to add that to your list.
It was in the first list, but it wasn't in the second list. That's true. Yes. No, you're absolutely right. Actually, this lady was the one who ordered the diagnosis, the molecular diagnosis. So she ordered it. She took it to her doctor, and he refused and gave her the existing traditional ones. And she did, of course, change her doctor. But I was personally shocked that the doctor had run out of options and refused to consider a proposal from molecular profiling. And this is, by the way, this is a fairly educated clinician. This is not sort of a jobbing oncologist from the backwards. Anyway.
Any other questions? Ernst?
But just a comment, because this again very clearly shows what the informed patient can do. And again, if this is not an incident that you happen to report to us, but that is actually something that the patient reports in some kind of information system to the world, it doesn't, as Jamie just said, it doesn't need a lot of patients to do that, to change the system. Because, and that, I think that is really the power of these molecular medicines, that you can inform the patient, that the patient, it's in his own best interest to do that. And so to change the system. So in all these figures, I think the patient has a much more prominent role than it had in the past.
Absolutely, yeah. If you say, I mean, I completely agree. If you say which groups of that lot are going to drive change, it is going to be number one, the patients, and number two, those of us that provide alternatives to those patients. It's got to be lunchtime, hasn't it?
It's almost lunchtime. No more questions? Okay, well, we can break. Thank you all very much. Thank you, Jonathan. And we're back here at 2.